Kristen Atchison here, and we're still talking about Chapter 13, the chemical senses, perceiving odors and tastes. We're in our third lecture, where we're going to talk about tastes and flavors. So we've already talked about olfaction in the previous two lectures. Now we're going to talk about gustation. And we did talk about a little bit that we mentioned that the sense of smell really does affect our sense of taste. So if we lose our sense of olfaction, it really does impact our sense of gustation. And this really has to do with the relationship between taste and flavor. So taste and flavor are different. Taste is the stimuli for taste. And these are tastes, we call them tastants, and they're just molecules, just like we called odors, odorants. We're calling tastes tastants. And these are molecules that can be dissolved in saliva. So just like we had a molecule detection for olfaction, we're going to have a molecule detection for gustation. And that's again why these are called the chemical senses, because it is this molecule basis for both taste and olfaction. So, researchers agree on the tastants, sweet, sour, salt, bitter, and umami. And there are other tastants that aren't agreed upon yet. There's some evidence that they think that the next taste will be fat, or they may give it another name, but that there is some evidence that there are other tastants. When I was a kid, we grew up with sweet, sour, salt, and bitter. The fifth one, umami, is a newer one. And so again, there is evidence that there may be other tastants added, but there isn't a consensus for that, and so that's why it hasn't been done. Flavor, however, is different. Taste is not the same thing as flavor. Smell is the main contribution to flavor. So again, when you have a cold or when you hold your nose, that's why that flavor changes, is because olfaction is a component of this. Other contributions are included in flavor as well. This will include some of the tactile properties, such as texture and temperature. Vision, how our food looks, is really, really important to us. Audition, how our food sounds, is actually important to us. In fact, the Braincraft video that I assigned, Taste with Your Ears, really talks about this role of audition in flavor. So make sure you watch that too. They do a really good job of describing studies that have done to see this role of these other senses in terms of flavor. So this is, again, flavor will be an example of cross-modal perception, because a lot of different senses are being incorporated to do flavor. So just like we've had absolute detection thresholds for olfaction, we're going to have absolute detection thresholds for tastants, for gustation. So again, here are the five: sweet, salty, umami, bitter, and sour. And the table does a really good job of listing some different substances that can cause this kind of response. So sweet can be caused by sucrose, which is just your typical table sugar, fructose, which is fruit sugar. It can also be used as a sweetener. Glucose, which is kind of our biological energy source. It talks about salty. It talks about umami. So things like soy sauce are that have that umami. And again, some of that has to do with that MSG that's in it. Bitter, quinine, that's in tonic water. So that would be a bitter, and it talks about this absolute detection threshold. Now remember, absolute detection thresholds are not being able to taste it, but being able to taste it and say what it is. So say that it's sweet versus salty. So it's not just detecting something, it's detecting something and identifying it. And so we'll see that there's different levels, and again, we're doing parts per million. Here it's being done in water instead of air, but we'll still see these differences. You can see that we're very, very sensitive to bitter taste. You don't need a lot of bitter for us to be able to notice. Where we aren't as sensitive, we do need a lot more sweetness or a lot more saltiness to be able to detect over some of the bitter ones. Again, that's going to be differences in what's important. Bitterness usually tells the system, this isn't a good idea. This is an unpleasant taste. Bitter is not something we typically like. And it can tell us, you shouldn't be eating this. So again, this can be a warning system. So we're going to be more sensitive to bitter things than we are to sweet things. So this is a nice image from your book that talks about kind of the relationship between taste and flavor, and how taste does not equal flavor. So flavor is the big blue box in the middle, and notice that everything's pointing to flavor. Our hunger or thirst, so kind of our motivation, other senses, including vision, audition, olfaction, we are contributing, as well as taste. Now, taste is broken apart into several different pieces too. We've already talked about the quality of taste, the salt, the sweet, umami, sour, bitter, but these can also be either pleasant or unpleasant. And we'll see that sweet, salty, and umami are seen as pleasant. Sour can go both directions, depending on if it's mildly sour or strong or sour. And we'll see that there's going to be a lot of individual difference there. And we'll see that bitter is usually seen as unpleasant. Taste also is going to include intensity of the taste. It's going to include location of the taste, and it's going to include kind of the onset and the aftertaste. So something may taste good initially, but we've all had that thing where there's that aftertaste and it doesn't have necessarily a pleasing aftertaste. So all of those things are incorporated in taste. And again, they are all a component of flavor, again, with those other senses modalities. So the anatomical and neural basis for our taste and flavor perception, again, make sure you've watched that crash course video. It does a good job of talking about how both taste and olfaction go in to contribute to flavor. So our taste buds house our taste receptor cells, or TRCs, and they are found in several other areas. Most of our taste buds are located on our tongue. However, there's a third of our taste buds that aren't on our tongue at all. We have them on our soft palate, we have them on our epiglottis, and we have it on our upper esophagus. So, you can sometimes get that bad taste right in the back of your throat. Those are those taste receptors in the back of your throat, the upper esophagus, that are being impacted. So again, our taste buds are not just on our tongue, but are also in other portions of the mouth and throat as well. Our taste receptor cells respond best to one type of taste versus multiple types of taste. So they're a little bit more similar to olfaction that way, but they're not exactly like olfaction where they're only responding to one. They're going to be more analogous to the color reception system, where they might respond best to one color wavelength, but they can still respond to other color wavelengths. TRCs only live for a week. So this is a really turnover kind of a system. And they're replaced by new receptor cells that are developed from these basal cells within our taste buds. So while you don't need to know the different kinds of taste buds and the different kinds of structures on our tongue, it is important to kind of see how this works. So our taste bud really is made up of a bunch of different kinds of cells. Again, the different kinds of cells aren't important. How it's being transduced isn't going to be important for this class. We're not going into that level of detail. But again, we have different kinds of structures in our mouth, and the taste buds are in different locations. And again, they're being made up of a multitude of different kinds of cells. There are two theories on how we process taste. In terms of how that information gets to our brain. One of them is the label line pattern. So these taste buds and these taste receptor cells are going to be attached to the cranial nerves. And the first theory says that there's individual fibers within that cranial nerve that are for each taste profile. So there's a cranial nerve fiber that's for sweet. There's a cranial nerve fiber that's for sour. There's a cranial nerve fiber that's for umami. This is called the label line pattern. That each line, each cranial nerve fiber, which is a line, is labeled for a particular taste. So for only one taste. The other theory is that we have an across fiber pattern, which is saying that these different nerve fibers are responsible for information from several tastes, so that it's across multiple tastes that we're going to, these cranial nerves are going to carry information for more than one taste. Now, neither one has been completely debunked, so we're still teaching both. However, there is a lot more support for the across fiber pattern model. There's lots of different studies that have looked at this and have found support for this. And your book does a good job of talking about those in some detail. You don't need to know the details of them, just that there is more support for the across fiber pattern. As I said before, those cranial nerves are carrying signals to the brain. Both agree on that. Both agree on how that information is transduced. There are 3 different cranial nerves that are sending information to the brain. We have the cranial nerve 7, which is important for our facial. It takes up the front 2/3 portion of our tongue and our soft palate. We have cranial nerve 9, which does the back third of the tongue. And we also have cranial nerve 10, which does the upper esophagus and the epiglottis. Now remember, both of those areas contain taste buds as well. They taste these taste receptor cells as well. So we have 2 cranial nerves that are working for the tongue, and then we have a cranial nerve that works for that upper epiglottis and the esophagus. And you can see how they've kind of broken that apart. So the pink part of the tongue is going to be cranial nerve 7. The yellow back part of the tongue is going to be cranial nerve 9. And the yellow part of the epiglottis and the esophagus is going to be cranial nerve 10. So the information leaves those taste receptor cells, and it goes through these cranial nerves to the nucleus of the solitary tract. Okay, this is in the brainstem. The information then is going to go to the ventral posterior medial nucleus of the thalamus. So again, olfaction is the only system that is not going through the thalamus. We have here, gustation is going through the thalamus. From there, this information leaves and goes to the primary cortex, which includes both the anterior insular cortex, which is highlighted here, and the frontal epiloecum. Then the information goes to the orbitofrontal cortex, the amygdala, and the hypothalamus. Again, we really just highlighted the orbitofrontal cortex here, but the amygdala and the hypothalamus are here, are indicated in this image as well. And information is going to those locations as well. We have individual differences in our taste perceptions, and these can be due to a number of different factors, one of which is genetic. We see a lot of genetic variability in the ability to taste bitter substances. And again, we'll see the differences in the behavior being linked to the differences in the genetics. We also have a genetic predisposition for preferences. One of the most well-known ones is cilantro. So some people really hate cilantro. They talk about it as having a soapy taste, and some people love cilantro. And they found that there is a genetic difference that's causing this. They have olfactory receptor genes in chromosome 11. This is specifically OR6A2, which you don't need to know, but if you're interested, there it is. And what they think is happening with this gene is that it's causing differences in the olfactory receptors, which is causing different responses, which is causing different preferences, either for cilantro or against cilantro. We also see that experience can play a role too. Things that you've talked about, I'm sure, in your intro classes and other classes, such as learned taste aversion, your own personal experience of things you no longer like because if they've made you sick, would be learned taste aversion. Previous experiences of foods that you like, foods that you don't like, cultural experiences, the flavors of your culture. Again, we talked about how that amniotic fluid is the flavor changes depending on what the biological mother's eating. So if the biological mother is eating the foods of her culture, the fetus is being given exposure to the flavors of that culture, and that experience of that taste profile starts prenatally. And so again, experience will play a role in this individual taste perception in terms of preferences, in terms of abilities, these sorts of things. So in terms that people have these genetic differences to be able to taste bitter, there are different classes of responses to being able to taste bitter. We have super tasters, and this is about 25% of the population, and they're going to be able to taste this better. They're going to have better detection than other individuals. We have tasters, which is the majority of the population, it's about half, and these can taste them. And then we have non-tasters, individuals who can really only taste these bitter substances at very, very high rates. And this is, again, makes up of that last 25% of the population. And they can see that this is due to differences in genetics. There is an exercise that is posted on Canvas for you to do, where you can kind of look at whether you are likely to be a super taster or not. This is not required. You do not have to do this. But if you're interested, it gives you a step-by-step way to kind of go through and see in terms of how many taste receptors you have at different locations on your tongue. Other examples of experience for taste perception has to do, again, with memory. So we see when we talked about kind of the system, the route that taste information was going in the brain, it was including areas for memory. We see that this starts prenatally as well. 5-month-old infants they tested 5-month-old infants, and what they did is they had their mothers in the last months of pregnancy. They had half the group drink carrot juice every day, and then we had half the group not drink carrot juice every day. And then when these infants were 5 months old, they tested the infants and asked and basically gave them a preference test, like Coke versus Pepsi, only this would be carrot juice versus something else. And what they found was that the 5-month-old infants whose mothers drank the carrot juice while pregnant, and carrot juice has a very strong flavor, so this flavor was being transferred to that amniotic fluid, preferred the carrot juice over the infants who did not have those previous experiences. So again, just like we saw memory playing a role very, very early on in development in terms of olfaction, we're going to see the same thing going on in terms of gustation. And what's interesting is, again, it's going to influence those preferences. Children that had been presented with those flavors were more likely to prefer those flavors than children that had not. So again, this is going to be kind of similar. We'll see that both taste and olfaction are really, really strong systems, especially at birth. We're going to see that these systems are online before birth. And again, this provides evidence for that. So this ends our conversation about taste and our conversation about the chemical senses. Thanks.